Picture this. Roughly 7 million years back, something incredible started happening. No drama, no big bang, just a quiet, gradual change that would eventually split our ancient relatives from chimps and bonobos forever. There wasn't some magical turning point where everything changed overnight. Instead, across thousands and thousands of generations, the very first early hominins slowly came into being. But here's what we really want to know. Who were these original humans that started it all? What did they actually look like? And how on earth did they make it in such a wild, dangerous world? If we want real answers, we've got to rewind the clock, way back, millions of years into the past. We're talking about our most ancient relatives, the true trailblazers of humankind that history nearly forgot. Our journey kicks off at the absolute beginning of our evolutionary story, deep in the heart of prehistoric Africa. This is where it all started, where the very first members of the human family took their earliest steps. And now, we're about to meet the oldest known species in our family tree. Sahelanthropus, chadensis. Meet Sahelanthropus chadensis, widely believed to be one of our earliest relatives. This species roamed west-central Africa somewhere between 7 and 6 million years ago. All the fossils we've discovered come from one location, Toros Manala, tucked away in Chad's Jurab Desert. What did researchers find? A few jawbone fragments, several teeth, and one remarkably well-preserved skull. The locals gave this skull a beautiful nickname, Tumai, which translates to hope of life. Here's the thing, we only have skull material from Sahelanthropus, no body bones yet. But even from what we've got, scientists can tell this creature was a fascinating mix. It had features from both apes and humans, though honestly, it leaned more toward the ape side. Let's break down the ape-like qualities first. The brain was tiny, estimated between 320 and 380 cubic centimeters. That's actually smaller than a modern chimp's brain. The face sloped forward, massive brow ridges jutted out above the eyes, and the skull stretched back in an elongated shape. But here's where it gets interesting. Sahelanthropus also had some surprisingly human-like traits. The canine teeth were small, and the middle section of the face was short, not drawn out like an ape's. The tooth enamel thickness fell somewhere between modern apes and a later group called Australopithecines, who lived from about 4 to 1, 9 million years ago. This suggests Sahelanthropus was in a transitional phase. One crucial detail: there was no gap, what scientists call a diastema, between the lower teeth. Most primates have this space, usually between the canine and the tooth next to it. But Sahelanthropus didn't. That's another sign pointing toward later hominins. Even the back teeth, the molars, were closer in size to Australopithecines than to great apes. All these dental clues tell us something important about diet and evolution. Bottom line? Scientists believe Sahelanthropus ate mostly plants, leaves, fruit, seeds, roots, nuts, and probably insects too. Now, here's a big question. Could Tumai walk upright? We're pretty confident the answer is yes, and here's why. There's a large opening at the base of the skull called the foramen magnum. It's where your spinal cord connects to your brain. In Sahelanthropus, this opening sits farther forward underneath the skull than it does in apes or any other primate except humans. That forward position means the head balanced on top of an upright body, strongly suggesting two-legged walking. However, and this is important, we haven't found any leg or foot bones yet, so we can't say for sure how well it walked on two legs. Most scientists think Sahelanthropus was probably a tree climber that also walked upright sometimes. Picture it walking on two legs between trees on the ground, but still spending most of its time climbing on all fours up in the branches, kind of like modern chimps, but with a posture better suited for standing tall. As for tools or culture, no direct evidence yet, but it's possible Sahelanthropus used simple tools like modern chimps do, unmodified stones, sticks, or easily shaped plant materials. The ancient world where these fossils were discovered was diverse and lush. Think lakes, forests, rivers, and wooded grasslands. Sahelanthropus shared this landscape with an incredible cast of animals. Researchers have uncovered thousands of vertebrate fossils at the site: elephants, giraffes, antelopes, hippos, crocodiles, lizards, monkeys, fish, and wild boars. Now, where exactly does Sahelanthropus fit on our family tree? That's still debated. One idea suggests it could be a common ancestor of both humans and chimps, a crucial missing link. Another theory says it's related to both lineages, but isn't a direct ancestor of either, meaning the evolutionary tree branches in more complex ways than we thought. And here's a wild card. Some researchers even propose Sahelanthropus might actually be an ancestor of gorillas, which would completely shift how we view this species. Regardless of where it fits precisely, Sahelanthropus chadensis, our friend Tumai, represents a profound moment in human evolution. Positioned near the very roots of our family tree, Tumai gives us rare, precious insight into those earliest steps on the path that would one day lead to us. Why they left the trees. Before we dive into other early hominin species, we need to answer one massive question. What made these early ancestors leave the safety of the trees and start spending more time on the ground? This wasn't just one thing. It was a combination of changing environments and genetic shifts. These factors pushed one lineage toward ground life and eventually walking on two legs, while the other stayed mostly in the trees. For a long time, scientists thought bipedalism, walking upright, first appeared on the African savanna. But newer research is flipping that idea on its head. It might have actually started in the trees. Studies on modern chimpanzees show they frequently walk on two legs while up in trees, especially when reaching for food. This challenges the old notion that walking upright was purely an adaptation to open grasslands. Maybe life in the trees played a bigger role than we realized. Walking upright in trees might have started as a way to reach food on branches more easily. Sure, moving into more open habitats definitely influenced how bipedalism evolved, but the research suggests those tree-dwelling habits gave bipedalism a head start before the landscape even opened up. So what changed the landscape? Around 6 to 8 million years ago, right when our ancestors were splitting from chimps, Africa went through major environmental shifts. Dense forests gave way to more open woodlands and grasslands. Why? Earth's climate was cooling down and drying out. This had to do with changes in ocean currents and dropping atmospheric CO2 levels. It was part of a long-term cooling trend that kicked off during the Miocene epoch. As temperatures fell, rainfall decreased across many regions, including Central and Eastern Africa. Tropical rainforests need lots of moisture. When that moisture disappeared, the rainforests shrank. In their place came woodlands, bushlands, and savannas. Walking on two legs offered some serious advantages in these new open environments. Better visibility over tall grasses, more efficient travel across long distances, improved temperature regulation, easier foraging, the ability to carry tools, food, and babies over millions of years. These habitat and movement changes drove deeper genetic and physical differences between the human and chimpanzee lines. But here's the thing. The split wasn't clean. It was gradual. There may have even been some interbreeding between the two lineages during those early stages of separation. Orrorin and Ardipithecus, the second oldest human ancestor after Sahelanthropus, is believed to be Orrorin and Ardipithecus. It's the only species in its genus, and these fossils date back to about 6 to 8 million years ago. So far, researchers have found 20 fossils from 4 different sites in Kenya's Tugen Hills. What do we have? Pieces of lower jaw, part of the chin area, several individual teeth, 3 thigh bone fragments, part of an upper arm bone, a finger bone, and the tip of a thumb bone. Orrorin probably still looked pretty chimp-like overall, but natural selection was already at work, slowly shaping a body that showed early hints of the modern human form. The thigh bone is the smoking gun for bipedalism. The rounded femur head, the long neck, and a key muscle attachment called the lesser trochanter pointing inward. All these features are linked to walking upright. But other bones suggest Orrorin still climbed trees too. One finger bone looks surprisingly human-like and has been connected to tool use. though in this case it probably had more to do with strong gripping for climbing. Brain size? Likely similar to a modern chimpanzee. But without skull material, it's tough to say for sure. Orrorin had relatively small teeth for its body size. The canines were still ape-like but smaller, similar to those in Miocene apes and female chimps. But the back teeth were small like modern humans. Orrorin lived in dry evergreen forests. This adds more evidence that bipedalism might have evolved in wooded environments, not just grasslands and savannas. Based on the low rounded molars and small canines, experts believe Orrorin ate mainly plants, leaves, fruits, seeds, roots, nuts, and insects. Like Sahelanthropus, Orrorin sits at the base of the human family tree but has more ape-like than human-like features. Some experts even think this species might have lived before the ape and human lines split, meaning it could be an ancestor of both, or it might be a basal member of the hominin group, basically an early offshoot. Ardipithecus kadabba, another hominin species from around the time of the human-chimp split, belongs to the genus Ardipithecus. Two species are described: Ardipithecus kadabba, dated to roughly 5-6 million years ago, and Ardipithecus ramidus, dated to about 4 million years ago. Ardipithecus kadabba comes from 11 specimens found at 5 locations in eastern Africa, specifically the Middle Awash region of Ethiopia. Discovered between 1997 and 2000, these fossils represent at least 5 individuals. What did researchers find? Teeth, jaws, hand bones, toe bones, arm bones, and collar bones. From these fossils, we know Ardipithecus kadabba was similar in body and brain size to a modern chimpanzee, but it walked on 2 legs. The canine teeth looked like those in later hominins, but the front teeth still jutted forward like an ape's. One bone from the big toe is broad and robust, suggesting it was used for pushing off the ground while walking upright. Here's something interesting. Unlike chimps, Orrorin and Sahelanthropus, who mostly ate fruit and soft leaves, there's evidence Ardipithecus kadabba ate a variety of tough, fibrous foods. How do we know? The back teeth are larger than a chimp's, but the front teeth are narrower. This suggests most chewing happened in the back of the mouth, perfect for processing hard-to-eat stuff like fibrous nuts. The environment where these fossils were found was a mix of woodland and grasslands with lakes, swamps, and springs. The fact that this species lived in both forest and open grassland environments complicates things even more. Did bipedalism evolve to take advantage of new open grasslands, like scientists once believed? Or did it first evolve in the trees? Or could both environments have shaped it? Scientists suggest Ardipithecus kadabba directly evolved into Ardipithecus ramidus, a species that lived about 4 million years ago. Ardipithecus ramidus is known from a partial skeleton nicknamed Ardi. This species gives us an incredible window into early human evolution. Ardi's bones, including parts of the skull, teeth, arms, pelvis, legs, and feet, reveal once again a species blending traits from both apes and later humans. The main features linking Ardipithecus ramidus to later hominins include small, blunt canine teeth with little size difference between males and females. This hints at a behavioral shift, possibly less aggression and competition. The skull was small-brained, like a chimp's, but lacked the powerful jaws seen in later species adapted for heavy chewing. It also didn't have the large front teeth used for eating tons of fruit, like chimps have. And once again, that forward-positioned opening at the base of the skull points to upright posture. Ardi's pelvis tells an interesting story too. The upper part showed clear signs of bipedalism, built for upright walking. But the lower pelvis and limbs? Still perfectly designed for climbing. This suggests Ardi spent a good chunk of time up in the trees. The shoulders sat high, great for reaching up to grab branches. This adaptation worked perfectly for climbing and hanging, unlike modern human shoulders, which sit differently to support long-distance walking, throwing objects and weapons, and using tools on the ground. Ardi also had long arms, long curved fingers, flexible wrists, and a thumb that wasn't very specialized. In other words, poor opposability. Orangutans are the only great apes with a similar thumb structure, and they also have poor manual dexterity. Ardi's feet had a big toe that stuck out to the side, just like a chimpanzee's, perfect for gripping branches. Clear evidence of a tree-dwelling lifestyle. However, there was also a unique small bone inside a tendon that likely gave more strength and stability for walking upright, and the hands lacked the special features needed for knuckle walking. This suggests Ardi was a transitional form, caught between tree life and ground life. This unique mix shows Ardi could move through trees but also walk on two legs, though definitely not like we do today. Fossils of plants and animals found alongside Ardi paint a picture of the environment. This species lived in relatively moist, heavily forested woodlands. The plant fossils include figs, palms, and hackberries. Animal fossils include columbine and baboon-like monkeys, kudu, peafowl, bats, shrews, rodents, doves, eagles, owls, and parrots. Ardipithecus ramidus was probably more omnivorous than modern chimps, based on tooth size, shape, and enamel. They fed both in trees and on the ground. Their diet likely included nuts, fruit, tubers, insects, small vertebrates, and eggs. The evidence isn't entirely clear, but studies suggest the front teeth were regularly used for clamping and pulling, possibly pointing to a diet that also included lots of leaves. Carbon isotope studies of the teeth reveal they ate woodland plants rather than grassland plants. This confirms Ardipithecus lived and foraged mainly in wooded environments, not open grasslands. The discovery of Ardi was a game-changer in the scientific community. Before this find, most people reasonably, but incorrectly, assumed the last common ancestor of chimpanzees, and humans looked a lot like a modern chimp, a fruit-eating, knuckle-walking primate mostly restricted to African tropical forests. But the Ardipithecus fossils don't have any specifically chimp-like features. This means the earliest hominins never went through a chimpanzee-like stage. This finding shocked many scientists and much of the public who had long imagined that human ancestors were basically early chimpanzees. Australopithecines and footprints after Ardipithecus. The next major group to appear in the fossil record is the Australopithecines. This group included several species, like Australopithecus afarensis, made famous by the Lucy fossil, along with at least 8 others. We know them from fossils found at numerous sites across eastern, north-central, and southern Africa. The various Australopithecine species lived from about 4 million to 9 million years ago. They had a more gracile, meaning slender and less robust, build than their predecessors. And here's the big deal. They're the earliest hominin ancestors that show definite, habitual evidence of bipedalism. Now, while the Australopithecines were definitely bipedal, many scientists believe tree climbing was still one behavior probably retained by all early hominins, including Australopithecines, until the first Homo species appeared around 5 million years ago. One of the most remarkable pieces of evidence comes from a set of footprints found in Laetoli, Tanzania. These footprints show where early hominins once walked along the shore of an ancient lake roughly 6 million years ago. The footprints were preserved in volcanic ash and most likely belonged to Australopithecus afarensis. They were made by 3 individuals, with one walking directly in the footprints of another, which made the original tracks tough to spot. These footprints prove the hominins walked upright habitually. There are no knuckle impressions whatsoever. For the first time in hominin evolution, we can see the feet had developed a modern human-like arch, rather than the flatter feet of apes and earlier hominins. The big toe lined up with the other toes instead of sticking out to the side. But the feet still kept some flexibility in the joints, and they had curved toe bones, suggesting they could still climb. Evidence suggests these hominins moved at a leisurely stroll. They may not have been capable yet of running at a faster pace. Australopithecus anamensis. Australopithecus anamensis, the oldest Australopithecine, existed about 4 million years ago. It had a mix of primitive features in the skull and advanced features in the body. The upper end of the shin bone shows an expanded area and a human-like ankle joint orientation, clear indicators of regular bipedal walking. Long forearms and certain wrist bone features suggest these individuals probably climbed trees as well. The elbow joint was more human-like in being relatively flexible, rather than the rigid locking elbows that quadrupedal apes have to support their weight as they move around. The skull combines some ancestral features, like a protruding face and a long, narrow brain case, with some more derived features such as forward projecting cheekbones. Australopithecus anamensis individuals had thick, long, narrow jaws with side rows of teeth arranged in parallel lines. Their strong jaws combined with heavily enameled teeth suggest they might have eaten hard, abrasive foods at times. But they were likely plant eaters in general, relying on both fruits and tough foods like nuts. There was an enormous difference between male and female body sizes, what scientists call sexual dimorphism. This size difference was similar to what we see in modern gorillas and orangutans. The strong physical differences between males and females, along with certain tooth features, suggest this species likely had a social structure involving multiple mating partners. There are two main possibilities. First, a polygynous system, where one male mates with several females, like gorilla groups. In this setup, both males and females usually leave the group they were born into. Or second, a polygynandrous system, where multiple males and females live and mate within the same group. This is seen in chimpanzees and bonobos. In their case, males stay in the group they were born in, while females leave when they become adults. If we use chimps and bonobos as a model for early human ancestors, the second option, a multi-male, multi-female system, is more likely. Plant and animal fossils, along with analysis of ancient sediments and rocks, provide clues about their environment. They lived near an ancient inland lake in the basin where Lake Turkana sits today. The region experienced volcanic activity. Various eruptions produced layers of volcanic ash that covered the ground. Fossilized remains of this species were found trapped between different ash layers, which has allowed scientists to reliably date these fossils to between 3, 8, and 4, 2 million years old. These early ancestors lived in forests and woodlands that grew near the lake. Australopithecus afarensis, Lucy. The Australopithecine species that came after anamensis was Australopithecus afarensis, Lucy's species, who lived between 3.9 and 3 million years ago, primarily in what's now Ethiopia, Kenya, and Tanzania. The Lucy fossil is one of the most incredible discoveries in paleoanthropology. It's a relatively complete skeleton, about 40% intact, that has helped scientists paint a much clearer picture of what these early human ancestors looked like. Many skull features were quite ape-like: a low sloping forehead, a projecting face, and prominent brow ridges above the eyes. The brain was still small, averaging around 430 cubic centimeters, about 1.3% of their body weight. But brain reorganization may have already begun, with some enlargement in parts of the cerebral cortex, which plays a role in thinking, memory, decision-making, sensation, and voluntary movement. A small hyoid bone, which helps anchor the tongue and voice box, found in a juvenile specimen suggests afarensis had a chimp-like voice box. This confirms they wouldn't have been able to speak. The semicircular ear canals were similar in shape to African apes, suggesting this species wasn't as fast or agile on two legs as modern humans. Their jaws and teeth fell somewhere between those of humans and apes. The jaws were relatively long and narrow. In the lower jaw, the teeth were arranged in rows slightly wider apart at the back than at the front. In the upper jaw, the placement of the last molar results in tooth rows that curve inward at the back. Sexual dimorphism still existed. Females grew to just over 1 meter in height, while males were much larger at about 1.5 meters tall. The pelvis and leg bones far more closely resembled those of modern humans and leave no doubt they were bipedal. Though adapted for walking rather than running, limbs displayed human-like features indicating the ability to walk on two legs. This includes thigh bones that angled inward toward the knee, arched feet with wide heels, and big toes that aligned with the other toes and weren't opposable. Ape-like features suggesting the ability to climb trees include powerful arms with long forearms, and shoulder blade sockets that faced upward like an ape is rather than to the side like a human is. But the shoulder blades did share other similarities with humans. The finger and toe bones are curved and proportionally longer than in humans. Most scientists consider this evidence that afarensis was still partially adapted to climbing trees. Others think it's just evolutionary baggage. However, the big toe appears to lack the grasping ability common to apes, and the hands are similar to humans in most other details. Relatively short fingers and a more refined thumb, which would have allowed for precise manipulation, though not yet to the same degree as modern humans. Microscopic analysis of tooth enamel shows they mostly ate fruits and leaves rather than nuts and other hard plant material. Their cone-shaped rib cage indicates they had large bellies adapted to a relatively low-quality, high-bulk diet. The position of the sagittal crest toward the back of the skull indicates the front teeth processed most of the food. This species lived in a wide range of environments. Some populations lived in dense forests, near lakes and rivers, while others inhabited open savannas or sparsely wooded areas. This ecological flexibility is significant. Australopithecus afarensis appears to be the first hominin in the fossil record to have adapted to life in the open savanna. Living in such varied habitats would have exposed them to a broader range of resources, encouraging dietary diversity, new foraging strategies, and possibly even behavioral shifts that laid the groundwork for later human evolution. Kenyanthropus platyops. Living in Africa at the same time as Lucy's species was another early hominin called Kenyanthropus platyops, who inhabited what's now Kenya. This relatively flat-faced, small bipedal species may actually represent a closer branch to modern humans on the evolutionary tree than Lucy's lineage. Before its discovery in 1999, the earliest known fossils with a flat face, a key shift in skull structure associated with modern humans, dated back only about 2 million years. Kenyanthropus, however, pushes that trait much further back in time. Its flat face could suggest a more direct connection to our lineage, possibly representing an early step toward the modern human skull. On the other hand, it's also possible Kenyanthropus was an evolutionary dead end, and that flatter faces evolved independently in multiple lineages. Others in the scientific community believe more fossil evidence is needed before we can confidently place Kenyanthropus within our family tree. The only known skull from the species was found badly distorted. These scientists believe Kenyanthropus platyops is nothing more than a variant of Australopithecus afarensis. In terms of brain size, Kenyanthropus was similar to Australopithecus afarensis, with an estimated cranial capacity of around 430 cubic centimeters. They had one of the longest known early hominin faces, measuring about 1–3 inches. Their nasal opening was narrower than that of Australopithecines, and their cheekbones were tall and sharply angled. Overall, the face resembles Homo rudolfensis, a species that came over 1 million years after Kenyanthropus. Due to limited skeletal remains, its overall body size is unclear, though it was likely comparable in stature to other early hominins. This species lived in an environment that mixed grasslands with wooded habitats, pretty similar to the sites at Laetoli, Tanzania, and Hadar, Ethiopia, where Australopithecus afarensis was found. While Kenyanthropus lived at the same time as afarensis, Kenyanthropus had smaller molars. This indicates the two species may have had different diets and therefore probably did not compete for the same types of food, though both were likely mostly plant eaters. Stone tools. Our closest living relatives, chimpanzees, along with other apes and monkeys, have been observed making and using simple tools. So it's likely all hominins use tools to some extent. But here's what truly sets us apart from other apes. It's not just using tools, it's modifying them, especially crafting and shaping stone tools. So that raises an important question. Did any of these early hominins actually create and use stone tools? So far, no tools have been directly linked to the very early hominins like Sahelanthropus chadensis, Orrorin or Ardipithecus. Beyond the simple lack of stone tool evidence in the archaeological record alongside these species, there's a big clue from the limited hand bones we've found. What we can gather is this. While their hands show some features that might have allowed them to handle simple objects, they likely lacked the specialized anatomy needed for the precise and forceful grips required to make stone tools. Instead, Their hand structure seems better adapted for climbing and tree life than for tool production. However, the Australopithecine species had hands well suited for controlled manipulation of objects. They probably did use tools. The oldest known stone tools are around 3 million years old and were discovered in Kenya. These Lomekwian tools, named after the site where they were found, were made from volcanic rock and crafted into flakes. A flake is a fragment of stone removed from a larger stone by striking it with another hard object or applying pressure. Researchers also found cores, the central piece of stone from which flakes are removed. Essentially, it's the leftover rock after flakes have been struck off. These provide valuable information about how humans made and used stone tools. Potential anvils were found too, heavy flat stones or rock fragments used as a stable surface to support other stones during the shaping process. Although Australopithecus afarensis is known from Kenya around this time, the most likely candidate for the toolmaker is Kenyanthropus platyops, since specimens of this hominin have been found close to where the tools were excavated. Still, don't count Australopithecus afarensis out. There's compelling evidence they may have been among the earliest toolmakers. A small number of animal bones found at Dikika in Ethiopia show what appear to be cut marks made by stone tools. They've been dated to about 4 million years ago, and the research team attributes the butchery to Australopithecus afarensis, as this is the only species known to live in the area at that time. However, the conclusions are controversial. If they hold up under scrutiny, this would be the earliest evidence of meat-eating behavior by a hominin. Australopithecus garhi, the first Australopithecine species to be strongly associated with stone tool use, is Australopithecus garhi, who lived around 5 million years ago. Fossils of this species were discovered at the Buri site in the Middle Awash region of Ethiopia. Archaeological evidence from the site includes antelope bones showing clear cut marks and signs of being broken open, presumably by stone tools likely to access meat and marrow. This suggests Australopithecus garhi may have been among the first hominins to transition to regular stone toolmaking and consumption of animal flesh, marking a significant step toward behaviors later seen in early Homo species. This species isn't well documented. It's defined based on one fossil skull and 4 other skull fragments. Although a partial skeleton found nearby from about the same layer is usually included as part of the Australopithecus garhi sample, judging by this limited sample, it was similar in appearance to Australopithecus afarensis, but probably slightly larger in body shape and size, with more advanced teeth features. However, the premolars and molars are larger than other Australopithecines and more similar to Paranthropus, a group of robust Australopithecines characterized by heavily built skulls capable of producing high stress and powerful bite forces. Garhi's brain size was about 450 cubic centimeters, similar to other Australopithecines. The associated fragmentary skeleton indicates a longer femur compared to other Australopithecine specimens. This suggests a shift toward longer strides during bipedal walking. However, long, powerful arms were maintained. A changing climate had thinned the forests that once dominated this region, and savanna grasslands were becoming widespread. Garhi most likely ate plant material, but if the antelope bones found at the site were indeed butchered by this species, then they must have included significant amounts of meat and marrow in their diet. Some scientists claim the large molar teeth show Australopithecus garhi is related to the Paranthropus branch. However, the combination of face and brain case features are unlike Paranthropus. The scientists who originally reported the finds think garhi may represent an ancestor of the genus Homo. Australopithecus sediba. The latest Australopithecine to live was Australopithecus sediba, who lived around 19 million years ago in South Africa. The fossil skeletons of sediba from Malapa Cave are so complete that scientists can see what entire skeletons looked like near the time when Homo evolved. Details of the teeth, the length of the arms and legs, and the narrow upper chest resemble earlier Australopithecines, while other tooth traits and the broad lower chest resemble humans. These connections indicate sediba may reveal important information about the origins and ancestors of the genus Homo. Functional changes in the pelvis of sediba point to the evolution of upright walking seen in Homo, while other parts of the skeleton retain features found in other Australopithecines. Measurements of the strength of the upper arm bone and thigh bone show sediba had a more human-like pattern of movement than fossils attributed to Homo habilis, a later hominin classified as human. These features suggest sediba walked upright regularly, and that changes in the pelvis occurred before other changes in the body found in later specimens of Homo. For example, many paleoanthropologists believe the evolution of the pelvis in hominins was driven partly by the increase in head size. The pelvis needed to accommodate the birth of larger-brained offspring, but reconstruction and analysis of two sediba pelvises revealed they had already developed certain modern features before brain size began to increase. This evolved primarily due to the demands of bipedal movement. The Australopithecus sediba skull has several derived features, such as relatively small premolars and molars and facial features more similar to those in Homo. However, despite these changes in the pelvis and skull, other parts of sediba's skeleton show a body similar to other Australopithecines, longer upper limbs, and a small cranial capacity. The most remarkable thing about sediba? They are believed to have walked with a fully extended leg and a unique form of bipedalism involving hyperpronation. This means when stepping, the foot would strike the ground on the outer edge, causing the foot to roll inward, with weight shifting to the inside of the foot before pushing off. While this gait was upright and bipedal, it differed from modern human walking. This suggests upright walking evolved along more than one path during human evolution. The combination of primitive and derived traits in Australopithecus sediba shows part of the transition from a form adapted to partial tree life to one primarily adapted to bipedal walking. Australopithecus summary. Although some differences occurred between the Australopithecine species, overall they were small, stocky hominins with a mix of ape-like and human-like traits. They had wide, shallow pelvises adapted for walking upright, though their rib cage and shoulders retained climbing features. Brain size ranged from 400 to 550 cubic centimeters, closer to chimpanzees than modern humans. They had pronounced brow ridges, sloping foreheads, and projecting faces. Their teeth fell somewhere between apes and humans. Smaller canines, thicker enamel, and larger molars suited for tough, plant-based diets. Long arms and curved fingers suggest some tree climbing, while their legs and arched feet supported bipedalism. They lived in a variety of environments, from woodlands and riverine forests to open grasslands, though rarely in fully open savannas. Fossil sites like Kubifora and Alia Bay in Kenya show they thrived in changing mixed habitats. Their diet was primarily plant-based, fruits, seeds, roots, and insects, with occasional scavenging of meat from predator kills. The genus Homo likely emerged from within the Australopithecines around 2 million years ago, but which species gave rise to humans remains debated. Fossils like Australopithecus sediba show transitional traits, more human-like hands and pelvis, suggesting a possible link. In summary, Australopithecines were key players in human evolution. Their upright walking, dietary adaptability, and early human-like features paved the way for the rise of Homo and eventually modern humans. Outro from Sahelanthropus, chadensis, and Orrorin to Ardipithecus and the Australopithecines. These early human ancestors lived across more than 5 million years, from around 7 million to under 2 million years ago. Slowly, through countless generations and gradual evolutionary change, they paved the way for the emergence of Homo. None of them knew the role they were playing. Each individual, each family, simply trying to survive day to day, most likely without speech. And yet their struggles and adaptations led to the humans who followed. Homo habilis, with their larger brains, dexterous hands, and long-term use of the Oldowan tool industry. The pioneering Homo erectus, who harnessed fire, crafted Acheulean hand axes, and became the first humans to spread beyond Africa into Asia and Europe. Then came the more familiar faces in our evolutionary story: Neanderthals, Denisovans, and of course, Homo sapiens, who would go on to build complex civilizations and develop advanced fields like mathematics, astronomy, and physics. Another important field, paleoanthropology, has allowed us to uncover the lives of these fascinating ancient hominins, and, in doing so, better understand the deep origins of our own species. All of us, every human who ever lived, can trace our roots back to those ape-like ancestors who slowly left the trees and embraced life on the ground, setting the stage for the rise of humanity.